Welcome to today's virtual event from the Jesus College Intellectual Forum. Through the year we run a lot of public events and we're planning to do some of these earlier on, but of course coronavirus meant that we couldn't have the audience we normally had. But we still want to let you know about some of the amazing work that's being done by people from Jesus College. So it gives me huge pleasure to introduce Matthew Young from the Wellcome Trust Sanger Institute and also one of Jesus College's postdoctoral associates, talk about the work that he's been doing on understanding childhood cancers.
Welcome everybody. My name's Matthew Young. I'm really excited to be here today and tell you about some of our work trying to understand the developmental origins of childhood cancers. So I should start by giving a disclaimer. None of what I'm talking about today constitutes medical advice. I'm not a doctor. To be honest, you'd have to work pretty hard to turn this into something that looks like medical advice, but nonetheless, I think it's worth saying. Furthermore, what I'm talking about here is fundamental research. I will touch upon some of the potential clinical applications of this work, but really the aim here is to understand the biology of the disease, understand the basics of development and how it goes wrong in childhood cancers. So with those disclaimers out of the way, I think it's useful then, well, it's necessary to begin by thanking the many people that contribute to this work. First and foremost, the children and their families who donated tissue, without which this work wouldn't be possible. And I really need to emphasize that this is a massively collaborative undertaking involving many tens of people, some of whose names are listed up there, and many institutions around the country and indeed around the world. So, to begin with, if we're going to understand the developmental origins of cancer, we need to know what a cancer is. So that's where we'll begin. At a fundamental level, what cancer is a disease of aberrant normal cells. Now, I'm showing you two slides here, one of a normal breast tissue and one of ductal breast cancer on this side. And even if you're not used to looking at images of cells under a microscope, I think you can probably appreciate that there's a pretty fundamental difference in the order and regularity of this side, where there are different structures and intermediate spaces filled with things that are not cells, versus this side, where one cell has just completely taken over, grown in an uncontrolled way, and in so doing obliterated the normal structure that the tissue needs to function. And really, this is what a cancer is. It's a normal cell that's grown in an uncontrolled way, and in so doing, taken over the tissue in which it sits and disrupted its function. So it's not just as simple as, in order for a cancer to survive and thrive, it needs to grow uncontrollably. That's definitely one of the things. And in order for that to happen, it needs constant stimulation, both from the cell itself and externally, from things called growth factors. It needs cues to continue to grow and proliferate. There's a nice analogy here to a strawberry plant where you don't just stick a seed in the ground and, well, maybe you do, and it grows. It needs many other conditions to be met in order for it to thrive. And in the context of cancer, some of the other examples are that it needs to evade attacks. In the case of strawberry plants, this is things like, bugs and birds and so forth. In a cancer context, this is fundamentally, this is essentially the immune system that needs to be avoided, which will try and come in and disrupt the cancer. And like strawberries need food and nutrients, so too does a cancer. It needs blood vessels that are coming in, providing nutrients and oxygen, and taking away waste factors. And without all of these different factors, a cancer really can't be successful. Now, I've said that a cancer is a disease of aberrant normal cells, and that's true, but we can go a step further than that. And to go that step further, we need to understand something about DNA, which I'm sure you all probably have an idea of what DNA is. But just to review, it's a book, sort of, is the analogy, I suppose, of 3 billion letters made up of A, C, G, and T, which is in every cell in our body, and it contains the instruction set, the information necessary to build a cell and tell it how to function. So what cancer really is a disease where that DNA in an individual cell has been changed in some way, and those changes to the DNA give it these properties that are necessary for it to become cancerous, to grow uncontrollably, to recruit blood vessels, to support its growth, and so forth. And usually this is, well, always, this is not just a single change of one letter out of 3 billion. What the picture really is that we start off as a fertilized egg, and as we go through life, changes to our DNA accumulate. They accumulate due to random chance. They accumulate due to exposure to environmental toxins like ultraviolet radiation and tobacco smoke. And most of the time, these changes don't do anything. That's what these little circles represented here are. Most of the changes you can induce to a genome don't have any actual effect. But occasionally, one of those changes has an effect, and that's what is represented by the stars here. And if you're unlucky, then those changes fall in just the right places, that they change the instruction set of our DNA such that the cell can go on and become a cancer. So really, at a molecular level, this is what a cancer is. It's a disease caused by changes to the DNA that change the biological function of a cell to have it behave in this terrible way. So to summarize what I've told you so far, cancer is a disease of the aberrant normal cells. Cancers need many properties to be met in order to thrive and grow. And the ultimate cause of cancer is changes to the DNA. So that's what cancer is. What is it that we're hoping to learn about through our work? So one other thing I want you to appreciate before I talk about what we're aiming to do is that the cells that exist during human development are special. They're fundamentally different from the mature cells of the adult body. And it's easy, I think, to appreciate why that would be if you're going to start off with an individual cell and end up with a fully grown baby in 9 months' time. The cells that make up that growing organism need to have the ability to grow and proliferate fairly rapidly and aggressively. And so the cells of development share many of the same properties that are defining characteristics of cancer. So I just want you to keep that in mind as we go forward. So the first thing we really want to ask is, and a lot of our work focuses on kidney cancers, and that's what I'm going to really talk about today. And when I say to you, we work on kidney tumors, you probably all have a sense that means that it's a tumor that arose originally from a cell of the kidney. But really what we want to be able to do is to go to a more fine-grained scale than that and say, well, we know it arose in the kidney, but if we look at a microscopic level in the kidney, what a kidney actually is an organ that's made up of this functional unit called a nephron that's repeated over and over again many, many times. Now, you don't need to know the details of all these different parts of the nephron or what their functions are, but just to appreciate that there are all these different parts They have very different morphologies. They're made up of different types of cells. And so a tumor that arises at this end of the nephron from the cells there is potentially going to look and behave and need to be treated in a very different way from one that derives at the distal end, at the other end of the nephron. And so we'd like to be able to take an individual tumor and match it to the normal cells of, in this case, the kidney, and see which one it most strongly resembles. Now, there's another idea that's been around in the literature for quite a long time, and this is this idea of childhood cancer as being a developmental disease. And the hypothesis is as follows. As I mentioned before, cells during development, a fetal cell, are different from mature cells. They have special properties. So, for example, you can have a cell in the developing kidney, which will, in the normal course of events, do its job. It will grow part of the kidney. But then, at a certain point, that growing of kidney is no longer necessary for the body, and so that cell will either die or it will transition to a mature state and lose that ability to grow a new kidney. And so the hypothesis goes that what characterizes some childhood cancers, if not all childhood cancers, is that there are changes that occur to the DNA in development, still in the womb, that cause those developmental cells to retain that ability to execute their developmental programs and continue to grow and proliferate after they should have shut off. And while this is a quite compelling hypothesis, there's not really any hard quantitative evidence that this is true or not true. So we'd really like to be able to have an attempt at quantifying this and testing this hypothesis. And there's a sort of mirror image to this hypothesis as well, which is that it's possible that, and it has been suggested, that what characterizes some adult tumors is that they revert back to this developmental state, that the changes to the DNA that they accumulate turn a mature cell into a developmental cell, and that's what's fundamental to driving the cancer behavior. And we'd like to be able to test this idea quantitatively as well. So the three key questions that we're trying to address here are, can we quantitatively test this idea of childhood cancer as having a developmental origin? Do adult cancers revert back to a developmental state? And can we identify for each individual tumor what is the normal cell that it most strongly resembles? So how do we go about doing this? In order to explain this, we need to go through some more background information. Now, this sounds a little bit intimidating, the central dogma of molecular biology, but I promise you it's actually not as complicated as it may sound. So we've already talked about DNA, this instruction book of 3 billion letters that's in every cell in our body. And there are special segments, parts of that DNA called genes, and what makes them special is they get turned into another intermediate molecule called mRNA. And this process of DNA being turned into RNA is called transcription. Really, I think a useful way to think about it is your DNA is really like a recipe book. In and of itself, it doesn't make a pizza for you. If you want to make the pizza, you need to take those instructions and act on them in some way. And for the DNA, the process of acting on those instructions is turning the DNA into RNA. RNA is not the end point either. It's another intermediate state. And what then happens is that mRNA gets turned into proteins, proteins being the small machines that make up the majority of the cell and drive its biological function. So the central dogma of molecular biology is just DNA contains genes, Genes get turned into RNAs, RNAs get turned into proteins, and proteins are really the things that are the determinants of biological function. They do the business that a cell needs to do. Unfortunately, it's technologically very difficult to very precisely measure all the proteins present in the cell. So instead, what we do as a proxy for biological function is try and understand which RNAs are present. Now, whenever I heard this story, I was always struck by something, which is that we have this picture that we have all these different cells in our body. They all have exactly the same DNA, but then you have a motor neuron and a white blood cell and a bone cell, and they all look radically different. They look radically different in terms of their morphology, they're radically different in terms of their behavior, and they're radically different in terms of their biological function. So if they all have exactly the same DNA, how is it that you end up with this immense diversity of cell types that make up our bodies? And the answer to this is, of course, that although they contain the same instruction set, they each use different parts of it. So the pattern of which genes are turned on and off and get made into proteins is different in a motor neuron, it's different in a white blood cell, and it's different in a cancer cell. And so if we want to understand at a molecular level what it is that makes a motor neuron and a cancer, what we need to be able to measure is the pattern of which genes are turned on and off. And that's what's shown here by this schematic. So each box is meant to represent an individual gene. I'm showing only 7 genes here. The real human genome has somewhere between 20 and 30,000 different genes. So you have to think of a version of this with 20,000 boxes, not 7. But the idea is still the same. Red boxes represent genes turned on, blue boxes genes turned off, and each cell has its own unique pattern of genes turned on and off, which define its biological function. So ultimately, this is what we want to be able to do. We want to be able to take a cancer, take the cells that make up the normal developing kidney and the mature kidney. We want to look inside these at a molecular scale and define this pattern of genes that are turned on and off and use that to understand how these cells behave at a molecular level. Now, I just sort of say in a sort of blasé way that this is what we want to do. Actually, that this is possible at all is sort of a miracle of modern engineering, biochemistry, and a whole intersection of different clever ideas and disciplines. So I think it's worth taking just a couple of minutes to go through a rough idea of how this actually is done. So what I'm showing you here is a schematic of what's called a microfluidics device. So these are tiny, tiny channels etched onto a chip, and you have fluids that flow through different channels of this chip that perform chemical reactions that are needed to measure the pattern of genes turned on and off in individual cells. So what we do is we take a piece of tissue, and a piece of tissue is comprised of cells joined together in matrix structures, and we treat it enzymatically, chemically, and try and break it up into a solution of individual cells floating around in a fluid. And once we've achieved that, we can flow that through one channel of this microfluidics device. So that's what these little colored circly things are here. And at the same time, we flow in a perpendicular direction a fluid containing these microgel bead things. Now, what these microgel beads are special little powerhouses that contain all the chemicals and biochemistry needed in order to do the process of working out which gene is turned on and off for a cell. So when these two things combine and get pinched off into a droplet, what you really have is a tiny mini laboratory within a droplet where these special chemical reactions take place, and we're then ultimately able to work out for each individual cell which gene is turned on and off. And the schematic's nice for understanding it, but I think it's also quite nice to see a video of how this actually happens. So this is just what I was talking about here. You see coming through these U-bends, these little microgel beads that contain the bits of chemistry needed for the reaction we're interested in. And you can see coming up through this channel here, every now and then, there goes a cell, there goes another one, and they join together and get pinched off into droplets where the reactions take place. So I'm going to tell you about really two large experiments that we've been working on to try and answer those questions of what normal cell does a tumor resemble, are childhood cancers developmental, and do adult cancers revert back to a developmental state? And as I said, our work really focuses on the kidney. So the first experiment, it works as follows. We have a kidney that's removed by a surgeon. We get a sample of tissue from the pathologist. We do this technique of trying to break it up into a suspension of individual cells floating in a liquid. We run it through this microfluidics device, and after a lot of hard work and processing, what that gives us is a readout of each individual cell of what is this pattern of genes that are turned on and off. It's transcriptional profile is another way of saying that, or transcriptome. And although we're interested in cancers ultimately, the starting point for all of this is to define what's going on in a normal tissue context. And this is where initiatives such as the Human Cell Atlas that we're a part of that aim to characterize at this transcriptomic level, this level of genes turned on and off, what's happening in all the cells of our body are incredibly useful. So first of all, we set out to define what's going on in the normal kidney in maturity, so that's post-birth in an adult, essentially. So here again is our schematic of the nephron, the functional unit of the kidney, and what we're aiming to do is take our piece of tissue, break all those individual cells out into just individual cells, and then get the pattern of what genes turned on and off in the glomerulus, in the proximal tubules, in the loop appendally, et cetera, And that's what I'm showing over here, not as a schematic, but this is actual data. This is an actual readout of which genes are turned on and off in individual cells in the mature kidney. And what you can see is that you can recapitulate all of the different key parts of the kidney that we know are there from previous work in our data. And this sounds a little bit circular in a way. We took a piece of tissue, we knew it contained many, many nephrons, and we knew the nephrons were composed of these different types of cells, and we then, lo and behold, were able to identify them. But of course, the power in all of this comes not from identifying these cell types are present, but that we now have an extremely precisely defined readout of, for all 20,000 genes, I'm only showing you 30 or so here, but we have this readout for all 20,000 genes in the human genome. What is the pattern of genes turned on and off that is defining of a glomerular cell in a normal kidney? And that's an extremely powerful tool that we can use as a reference going forwards. Now, as I said, we're interested in development and how that intersects with childhood kidney cancers. And so it's vital that we also do this not just in mature kidney, but also in the developing kidney. And the process here is essentially the same. You take a piece of developing kidney tissue, you dissociate it into individual cells, and you run the experiment. And the cell types that characterize the developing kidney are, of course, very different to those in maturity, but nonetheless, we can identify them and again get this fine-grained, really precise readout of what are the patterns of genes turned on and off that are defining of these different populations of the developing kidney. So now we can finally turn to our tumors, to our cancer tissue. And again, we're doing the same type of experiment. We take a piece of cancer tissue, we dissociate it, and we measure the patterns of genes turned on and off in those cells. Now, having talked about cancer as a disease of aberrant normal cells that grow in an uncontrolled manner, you might be tempted to think that this is a little bit pointless. You just be measuring a copy of that same cell repeated over and over again. But actually, that's not the case. What we find when we do the measurement is that, of course, we don't just capture the tumor cells, but we capture all of the other things that are necessary to support it. So for instance, we capture the cells that make up the blood vessels that the tumor has recruited in order to support its growth, and the entire barrage of infiltrating immune cells that are attacking the tumor to try and suppress its growth. So, but the process is then the same. We identify each of these populations of cells and get a readout of what is the transcriptome for the tumor cells, for the cells of the blood vessels, for the immune cells, and have a precise characterization of the genes turned on and off for each of them. So after all this work, this is really just the starting point for our analysis. What we're taking as an input are these reference maps of the normal kidney in development and maturity, and this gene expression pattern, the pattern of genes turned on and off in the tumor cells. And what we want to do, what we want to get out of it, is a measure of which of the normal cells, if any, does a particular tumor resemble. And we do this by constructing a mathematical model. I won't go through the details of this mathematical model, but the idea is that we train this model on the reference, the kidney population, and get a definition mathematically of what is the pattern of genes turned on and off that characterizes all of the different reference populations of cells. And that's what's shown in this heat map here along the bottom axis. These are the reference populations in the normal kidney. And then what we want to do is use this model to calculate a score of how similar are each of the cells in our tumor sample to the reference normal population. So on this vertical axis here are the cells that we're testing against this reference, the cells present in the tumor sample. And the color scale then tells you brown cells don't look anything like that particular reference, and green tells you that there's a very strong resemblance. Now, actually, most of this heat map is taken up by what we scientists love, which are controls. These are things that are telling us that the technique we're doing, the method we're applying is behaving in the way we would expect. And here the controls really are that, well, if our method is not telling us that the cells that make up blood vessels in the tumor look very similar to the blood vessels that are present in the normal kidney, then it's doing something wrong. If it's saying blood vessels look like immune cells, then something's way off base. And likewise, all the different components of the immune cells present in the tumor should largely resemble the immune cells present in the normal kidney. And all of these rows here are telling us essentially that the immune cell monocytes in the tumor look the most like monocytes in the kidney and not like any of the other populations. And indeed, if we put in an extra control population of cells from the adrenal gland, we find that they don't look anything like any of our reference populations, because the adrenal gland is a different tissue, it's made of different cells, and nothing in our reference looks like that. So finally, we come to the tumor cells, and we have confidence in our method for comparing cells in the tumor to our reference population, and we find that for this particular tumor, there's a very, very strong match to one particular type of cells of, in this case, the developing kidney. So this is extremely powerful and very important, and gives us the ability to precisely define, using these techniques, what normal cell a individual tumor most resembles. However, there are some disadvantages to this as well. And really, the disadvantage is that this approach is very expensive. And it's expensive in terms of money, but it's also expensive in terms of the logistical difficulty of executing these experiments. The tissue needs to be collected, the laboratory experiments need to be run, and they need to be run almost immediately after the sample is collected, because the RNA will degrade if it's not done immediately. And really, what we want is, it's very useful to be able to see what's happening for 1, 2, or 3 individual tumors, and that's the sort of numbers that are really viable. But really what we want to be able to do is to expand this to hundreds or thousands of different tumor biopsies to get a representative picture of what's going on both within different samples of the same type of tumor and across different tumor types of the kidney. And so that brings us to the second experiment I want to talk about. Now, as before, we do the same process to define what's going on in the normal kidney. And I really can't emphasize this enough. This whole approach just relies on having a very, very high quality reference, a high quality understanding of what's going on in the cells of a normal kidney at this, using this measure of genes that are turned on and off. But we only need to do this once, really. Once we've established this high quality reference, we don't need to regenerate it for each patient. So instead, what we do to expand this approach from a few individual cases to thousands of cases is to not get a readout of each individual cell that makes up a tumor, but instead use a different, cheaper, more established technique that allows us to take a piece of tissue and just get a readout of what's the average pattern of genes that are turned on and off across all cells that make up our tissue section. So this approach is called bulk transcriptomics, or bulk RNA sequence. Bulk referring to the fact that we're not measuring what's going on in individual cells, but the bulk of the tissue. So our task then becomes the following. We have this, and of course I should say that the advantage of this bulk approach is that it's established, it's cheaper, but also it's been around for the better part of a decade, and so there are thousands of samples that have already been run, tumor samples that have already been run using this approach. So we can take this data and immediately start getting insights into what's happening across large numbers of tumors. So the task is really the following. Our bulk data represents an averaging across all the individual cells that make up our piece of tissue, and that's what's represented by this smoothie here. It's a blending together of the flavor profiles, if you like, of the individual components that make it up. And expensive but very precise experiments allow us to define very precisely what the individual components that make a normal kidney look like, what a strawberry looks like, what an orange looks like. So what we aim to do is to construct a mathematical model that says, okay, well, we have this measurement of what the average pattern of genes turned on and off in the tumor is. And we have a high-quality reference that tells us exactly what the pattern of genes turned on and off in a strawberry is, a kiwi is, a mango, et cetera, et cetera, et cetera. And so we want to construct a mathematical formulation that takes those references, that it takes that bulk measurement and tells us, well, how much of that signal is coming from the strawberry, how much is coming from the kiwi, how much is coming from the mango. And again, I won't tell you the details of how we do this, but when we do, we get the following. So what I'm showing you here, each dot represents an individual tumor measured using this bulk approach. And the position on the horizontal axis, the blue, red, and different colored boxes, say how much signal is coming from different populations in our reference. So the blue represents all immune cells, the red represents the cells that make up blood vessels. And the displacement, the height at which this point lies, tells us the size of the contribution from that reference population. So what we're seeing here is a measurement of across tens of different tumors, all of the same type. These are all a particular rare subtype of childhood kidney tumor called congenital mesoblastic nephroma. And what we see for this tumor type is that there are three key populations that make a non-zero contribution to explaining the pattern of genes turned on and off. And they are that there's some level of immune infiltrate. There are immune cells present in all of these different samples. There is a network of blood vessels that supports these tumors. Neither of these things are surprising. The fact that these things come up give us, again, confidence that the method is working as we expect it to be. But then, of course, the one that stands out is this final population, which represents a particular type of cell in the developing kidney, which we then posit is the normal cell that matches the tumor cells in these bulk samples. And it's interesting to note that this is looking across 20 or 30 different tumors, all CMNs, and yet this signal is predominant in basically all of them. So despite there's diversity in the particular changes to the DNA that give rise to these different tumors, but despite that, we find a clear and consistent signal that each of these tumors matches the same population of normal cells in the developing kidney. And to validate that, we then do one or two targeted single cell experiments, so where we take the tumor tissue and measure the pattern of genes turned on and off for individual cells. And when we do that, we find that, lo and behold, it's the same population of normal cells that the tumor cells resemble. So this approach is really telling us what the normal cell correlate is for each of these tumor samples. Now, I've talked a lot about matching normal cells to tumor cells, essentially, but I mentioned two other aims, which were to quantify really the aggregate immaturity of a tumor sample. And this approach allows us to do that as well. And we do that in the following way. We can take our piece of bulk tissue that we've defined the average pattern of genes turned on and off for, and we can pretend that we don't know anything about whether it came from development or maturity. We'll just say it's a kidney tumor, it came from somewhere in the kidney, so we'll give our model the full suite of different reference populations. Every cell that's present in the developing kidney, every cell that's present in the kidney in maturity. And the model is then free to assign the signal to whatever population does the best job of explaining the pattern of genes turned on and off in the particular tumor. And what we then do is just add up the contribution from all of those references that come from the developing kidney, which gives us an immaturity score, a quantification at an aggregate level of the immaturity of that piece of tissue. So the first thing we do is to run some checks to see if this behaves the way we expect it to. Is it doing what we think it should be doing? And the way we do this is to calculate this score for bulk transcriptomes, so bulk measurements of genes turned on and off, from normal kidney tissue across the range of, across the human lifespan, really. And that's what's shown here. So the value on the horizontal axis tells you the age of the person from which the kidney was taken, and the vertical axis tells you the immaturity score that we calculate. And what you can immediately see is that all of the postnatal, the post-birth tissues, all have an immaturity score within this fairly narrowly defined range. You might see a hint that there's a sort of uptick going towards those kidneys taken just immediately after birth. That probably does represent something, some lingering developmental processes going on there, but nonetheless, they all fall in this fairly narrow range. And if we look at samples taken from during development, they have a much, much higher, statistically significantly different immaturity score. So this is what we expect. It gives us confidence that this does tell us something about the immaturity of a piece of tissue, and it gives us a range to define what normal immaturity is. We then apply this to our entire range of 1,000+ kidney tumors. And what's shown here on one side are the adult tumors, and on the other side, all the different subtypes of childhood tumors. And what's immediately striking is that systematically, all of the childhood tumors have a high immaturity score. At a statistically significant level, all of the samples that make up these, all of the samples that are taken from childhood tumors are immature. This is direct quantitative evidence of this idea of childhood cancer as a disease of stalled development, of ongoing immaturity. By contrast, the adult tumors have essentially no samples that have an high immaturity score. And this is perhaps surprising, right? Because there is this idea that adult tumors may be reverting back to a developmental state, and that's what is driving the disease. But what we see here is that actually at this zoomed out level of the whole transcriptome, the whole pattern of genes turned on and off, this doesn't appear to be the case for the vast majority of kidney tumors. There are, of course, interesting exceptions. The one that I've highlighted here as standing out particularly is a particular subtype of adult kidney cancer called chromophobe renal cell carcinoma. Now, usually chromophobe renal cell carcinomas have a better prognosis than the more common type of renal cell carcinoma, but there's this subtype of the subtype that has a higher immaturity score and is characterized by, unfortunately, almost being universally lethal, as seen there. And this is a pattern that we see in other ways in our data, that for an adult tumor, having a high immaturity score is usually pretty bad news. So to summarize the main points here, we can use this cutting edge molecular biology, molecular technique to measure the patterns of genes turned on and off in individual tumors and correlate that to normal cells, both of development and maturity. Using these techniques, we can provide quantitative evidence of this idea of childhood tumor as being tumors as being a disease of stalled development. And at least for kidney tumors, we find that most adult tumors do not revert back to a developmental state, at least at the level of the whole transcriptome. So I'll just finish with a final case study which touches upon some of the ways that these sort of insights may have some clinical utility at some point in the future. So this is a case of an 11-year-old that presented with a kidney tumor. Now, the most common type of childhood kidney tumor, Wilms tumor, has a peak age of incidence at about age 4. And so when a kidney tumor presents at this age, there's a question of, is this a late occurring childhood type tumor or an early occurring adult type tumor? And there's standard microscopic techniques that are used to try and make this diagnosis and differentiate which of these cases it is. But in this case, these results were essentially inconclusive, and the pathologists couldn't really agree on what the diagnosis should be, and weakly favored the diagnosis of an adult type tumor. And what we did was to run this sample through our analysis. So we had a readout of what the aggregate pattern of genes turned on and off was for this individual tumor. We calculated its immaturity score, which is what's shown here. And what we immediately see is that it has a higher immaturity score than we would expect for an adult type tumor, strongly suggesting that this is a childhood tumor. And in fact, if we dig further into the details of what exactly are the normal cell populations that explain the signal in this cancer, that further supports the idea of this being a childhood tumor. Now, it just so happened that for other reasons, I should say that all of this was not done before the diagnosis was made. This was done after the fact. And it just so happened that this individual was treated as if it was a childhood tumor, and the treatment protocol thankfully worked, and the child continues to do well, which really would only be the case if this diagnosis was correct. So I'll just leave you with that sort of final thought, which is that we've already established that understanding the changes to a DNA are an important clinical variable to measure as a part of cancer care. And this information that I've been showing you, this picture of exactly which genes are turned on and off, which really define the biological function of the tumor, can tell us a lot. Crucially, it's not information we can just infer directly from the changes in DNA. Knowing the changes in the DNA don't tell us what the patterns of genes turned on and off are. This technique is robust, it's cheap, and I hope that in the future we'll be able to utilize it clinically to help in these sorts of ways. Thank you, everyone. It's been a great pleasure to be able to tell you about our work trying to understand childhood tumors, and once again, thank you very, very much to all the many institutions and people that have contributed to this work.